Chemical pollution is an all too common story in aquatic environments. With the development of modern chemical engineering, we are able to manipulate compounds for just about our every whim. But with this abundance of chemicals, it inevitably leads to some slipping through the cracks and ending up in places where they don't belong. As of late, a compound known as 4-Nonyphenol, or 4-NP, has been gaining a lot of attention within the marine toxicology community because of its growing effects on marine organisms, namely its disturbing effects on finfish reproduction. 4-NP is a man-made organic compound found in common products such as detergents, cosmetics, paints, plastics, and birth control. It finds its way into marine environments in ways such as runoff, wastewater discharge, and most of all, sewage effluent. When used for contraceptive purposes, excess 4-NP is excreted out of the body in the form as waste. This waste is rich in hormonal pollutants known as endocrine disruptors, meaning it interferes with the body's ability to produce and regulate hormones. Once it finds itself in waterways is when it becomes a threat to fish populations. 4-NP is a lipophilic substance, meaning it likes to build in lipids and cell membranes of the body. This causes it to bioaccumulate in the tissues of fish. When the chemical can't be broken down, that's when it starts to affect the fish. 4-NP is an anti-androgen, meaning it mimics estrogen and takes its place and binds with estrogen receptors, effectively disrupting the male reproductive system in a process known as feminization, hormonally induced development of female sexual characteristics in male fish. Now, it isn't uncommon in the fish world for fish to swap genders at some point in a lifetime. Many species do it, such as clownfish, wrasses, groupers, and parrotfish. It's a reproductive strategy called subsequential hermaphrodism. It's meant to serve as a curve when numbers of a sex are running low. However, hormonal swaps are occurring in non-hermaphrodites because of 4-NP and other hormonal pollutants. This pollution creates feminization in species where it shouldn't be occurring. It poses a very significant and very real threat to fish populations. 4-NP exposure induces vitellogen production in male fish, also called VTG. Vitellogen is a hormone that's normally secreted from the female liver for oocyte production. However, VTG production in male fish inhibits gonad development, limiting reproductive success. This vitellogenesis is a hallmarker of 4-NP exposure and environmental estrogens. Juvenile African catfish were exposed to 4-NP concentrations of 0.3 milligrams per liter. These catfish showed deformity and fusion of semi-infernous tubules. These tubules are what keep sperm in production throughout the life cycle of the fish. With these deformities of these tubules, sperm production becomes scarce. Lower sperm counts are a red flag when it comes to reproduction. Fish generally reproduce via external fertilization, which is only a fraction as efficient as internal fertilization. Only a small percentage of released sperm makes it to fertilize eggs. With less sperm, less eggs will be fertilized and reproductive rates will drop, followed by population numbers as only a fraction of juvenile fish reach reproductive maturity. To make things more interesting, or concerning, 4-NP is also negatively impacting hermaphroditic species. Rockfish are known as protogynous hermaphroditic, meaning that a female rockfish could transition into that of a male, but it couldn't work the other way around. Rockfish exposed to 4-NP have increased vitellogenesis and have a decreased gonadic somatic index. With smaller gonads, there is less ability to produce and regulate hormones. Without adequate hormones, transitioning can't occur when it's needed, and male rockfish with small gonad sizes experience decreased sperm counts, driving a decrease into reproduction. When it comes to female fish, 4-NP still takes its toll. Medaka, also called Japanese rice fish, that are exposed to a concentration of 100 microns per liter, showed a decrease in egg production, and because of the bioaccumulation, the hatchability of these eggs were adversely affected, and the poisoning is carried on into the second generation. Because 4-NP bioaccumulates, once the effects take place on the endocrine system, it is unlikely that they will be able to be reversed. Sperm counts will stay low, and sex hormones will be skewed, and they will stay that way, possibly permanently. All of these factors serve to decrease the reproduction rate of finfish exposed to 4-Nonyphenol, putting marine populations and communities and ecosystems at risk. If population numbers dwindle in an ecosystem without much functional redundancy, it could cause an entire ecosystem to collapse.